Good morning. Today we're going to be talking about the introductory notes for Biology 103, or if you were in the hybrid course, Biology 103 hybrid. So the first part of this, we're going to get into kind of the foundation of science and the scientific method, and we'll have an activity on it. But there's two types of reasoning that can be done in science, and one of them is deductive and the other is inductive. So deductive would be if you use some general principle to explain some observation. So to do this, you might start out with a general statement and then make a prediction based on that statement. And we'll look at some examples. And just the word deduction literally means a conclusion drawn from a general principle. So we'll start with that principle and then we'll make conclusions from it. Whereas for inductive reasoning, the conclusion is drawn from observations. So we move from observations to conclusions, which is what an inference is. So for inductive, you should think of inference. For deductive, you should think of a deduction. And that's what they're based on, and we'll look at some examples. So one way to think of deduction would be flowing from the general, so some general statement about anything, to a specific conclusion, whereas inductive or inference would flow from specific facts or data to a general theory, perhaps. So an example of deductive reasoning, we could say, all dogs are mammals. All mammals have kidneys. So we just have these general statements. All dogs are mammals. That is true. All mammals have kidneys. Therefore, dogs have kidneys. So we come to a conclusion based on some general statements. Another example, this is deductive reasoning again. So since all squares are rectangles and all rectangles have four sides, so these would be the general principles or statements. So a conclusion we could draw from that is that all squares have four sides. And as long as the premises, the initial statements are true, then the conclusion will be true if it flows logically from those prior statements. For inductive reasoning examples, we might have an observation. You might think every quiz you've taken so far has been easy. So that would be an observation. So then a conclusion you might draw from that would be the next test will be easy. Or the test based on those quizzes will be easy. And in this course, your test will be largely based on those quizzes for each module, so make sure that you're keeping up with those. Don't wait till the last minute. Another inductive reasoning example, it says the bank was robbed and the safe was opened with the combination. So those would be the observations. And if only Jeff and John have the combination, then perhaps Jeff or John robbed the bank. So these are all observations the person has personally seen perhaps or heard, and then that leads to their conclusions. So inductive, think observations. For deductive, think of general statements. And that can help you remember when you're picking these out on a quiz or a test, which is which. And we'll do a test review, of course, before we get there. So scientific method is the next topic. Hopefully you're a little bit familiar with this. Basically, the general idea here is the scientific method is those general procedures and ways in which we approach answering questions. So it starts with knowing what the question is. So you ask a question, and perhaps, so for example, when I was in my undergraduate, we were studying plants in my research class. So what I did was, I had been watching videos about how fluoride was really bad for our health, and I went down this YouTube rabbit hole about that. So I was curious what fluoride would do to plants. So my question was, how does fluoride affect plants? So then you would construct a hypothesis, an educated guess about what you think the effect might be. Then you perform an experiment to test that prediction, to try to pursue answers to that question that you've asked. And for me, it was, how does fluoride affect plants? My prediction was that it would harm them in my hypothesis, and we'll get to how you write those hypotheses. And then I performed an experiment over the semester, and then you form conclusions. And actually, through my experiment, I determined that at least at the levels that I was treating the plants with, which was the same concentration of fluoride you would have in toothpaste and more, those plants actually did better. They they actually did completely fine. So one of my conclusions was that it does not negatively affect plants at those concentrations. Now, there are plenty of papers that if you do more than that, it does affect them negatively. So anyway, you would form your conclusions based on that experiment. And that scientific knowledge that we gain as a community comes out of that, following that method, asking the right questions, constructing predictions or hypotheses, performing an experiment, and then you base your conclusions on all of that evidence that you have through that process. And the great thing about it is it's repeatable, and that's really good for approaching problems. If I say fluoride's bad, someone else can repeat that same method and see if they come to the same conclusion. And if we both do, following the scientific method, then that's a good indication, if our sample size is right, we've controlled for all of our variables, that we have come to something that is true about the world. So we need to observe and ask questions, of course. And you've got to recognize what the problem is that you're trying to understand. So you're going to use your existing knowledge. With my plant experiment, there's a picture of, not my personal plants, on my new hard drive. I don't have the pictures of those original plants, but that's a picture of a plant that has been dipped in a fluoride solution. So you can go out and do some research to see what other people have found on that topic. So you should include existing knowledge. You should be referencing those papers that have already been written. And the observations when you do your experiment, they could be qualitative, right? For this plant right here, we can see that qualitatively, I can't numerically determine how yellow that leaf is, maybe with a computer program, but just generally, qualitatively, which means non-numerically, I could say that leaf is looking green or looking yellow, and that's a sign of poor health. Another observation could be quantitative. So what I did with my leaves was I crushed them up and turned it into a liquid and used a centrifuge to get the chloroplast out. And then we did a procedure called DC-PIP reduction, which all of that you don't need to know. But the point was, we did an experiment to measure numerically how fluoride had affected the plants. And what we measured was its ability to perform photosynthesis, the rate at which it could do that. So when you do that experiment, only thing to think about here or to remember is that you're either measuring numerical values, which is called quantitative data, or you're measuring non-numerical values that aren't easily measured with a ruler or a stopwatch, and we call those qualitative, right? They appear to be in good health. Then you want to construct your hypothesis. So a hypothesis would just be an educated guess or a prediction or explanation it should be based on some type of previous knowledge. And it has to be testable. If you have a hypothesis that is not testable and you can't collect data either qualitatively or quantitatively, then what's the point? You can't figure anything out from that experiment. So you want to make sure you're asking the right questions that can lead to testable hypotheses. And if that's what you're doing, then you're probably asking the right questions. Then you can construct that hypothesis, make sure it's testable. So for example, if I said, if we give plants fluoride, then they will die. And that's just a simple, crude example, but I can measure if they die. Perhaps a better example would be, if we give plants fluoride, then their photosynthetic ability will be inhibited. So then what are the variables in the experiment? Well, a variable, just definitionally, is any factor that can change in an experiment. So there should be only one variable that you're testing, and all other variables should be held constant or controlled for. For example, with my fluoride plants, not looking at this example here, but this example you could substitute juice there in the middle with fluoride perhaps. We want to make sure that the plant that gets fluoride, that's the only difference between the plant we're measuring and the controlled plant. So you want to give both plants everything exactly the same sunlight, the same water, the same temperature in the room, but one plant gets some fluoride dissolved in its water, whereas the other plant just gets water. That way you know if the variable you're measuring for is actually causing the change and not something else. If I give the variable, or if I give the sample fluoride but I don't give it as much water, and the other plant, the control group, gets more water, then you don't really know if it was the fluoride that harmed the plant or the lack of water. So you want to hold everything constant except that experimental variable. Now, with this, we have two types of variables. We have an independent variable and we have a dependent variable. So the independent variable, the way to remember this is the variable or factor that is being tested. It's being manipulated by the scientist, and they're trying to figure out what effect this thing has. So the independent variable is what is being tested, and the dependent variable is the response that you're measuring. So again, with the fluoride example, the independent variable would be what we're testing. So the value that we're testing, the independent variable, is the amount of fluoride we give to the plants. That's what's being tested, because we want to know how fluoride affects the plants. The dependent variable depends on how much fluoride we add to the plants. So if we add a lot of fluoride, well then the response that we measure dependently, it depends on the amount of fluoride. That's the dependent variable. So that might be how tall the plant grows, how healthy the plant looks. It could be quantitative or qualitative, what we're measuring. But the dependent variable is the response that we're getting. So here in this experiment, we have a plant that's getting a cola, plant that's getting some juice, and a plant that's getting some water. So this plant with the water, sunlight, controlled temperature, that will be our control group. Everything's constant. And then with our experimental groups, we're adding some substances. So here we could be testing the difference between Coke, juice, compared to our control group that's just getting water. And the constant would be everything that's held the same across all of our groups. So here they're saying the dependent variable is how tall the plant grows based on that substance that's being added to it. So here we could have two experimental independent variables. One variable is the juice, the other variable is the Coke. We could have another substance, fluoride. Then you've got three experimental variables that would all be independent. Those are the independent variables. And then the dependent variable for all of the groups would be the plant height. You need to make sure you have a true control group. Here's an example of an experiment being able to pick out the independent variable, dependent, and the experimental and control group. So it says an experiment for a new asthma medication was set up into two groups. We're not supposed to have the answers on there already, but we'll just go through it. So they're testing this new asthma medication, and they've got two groups. Group 1 is given the new drug for asthma, while Group 2 was given the traditional medication that asthma patients use. So they're going to compare the new drug to the original medication. The patients were then monitored for 6 months, so that would be a constant. During this time, or a control variable, during this time the number of asthmatic events were recorded, i.e. number of rescue inhalers required, emergency room visits, hospitalizations, etc. Group 1 saw a 25% reduction in the number of asthmatic events in comparison to Group 2. So the independent variable, that's what's being measured by the experiment. What are we trying to determine? What are we trying to measure the effect of? We're trying to measure the effect of this new drug. So the independent variable would be the new drug for asthma. What is the dependent variable? So what is going to depend on whether or not this person takes the new drug for asthma? That's going to be, whatever we're trying to measure. So what effect are we measuring? So one dependent variable would be the number of asthmatic events. So the independent variable is they're given the drug. That's what's being measured. And then we're trying to see what effect does that have. Well, it may reduce the number of asthmatic events. And in this case, it says they saw a 25% reduction in the number of asthmatic events. What is the experimental group? Experimental group is the group given the independent variable, the group given the new drug. And the control group would be group 2 because they were given the original drug. And then the constants would be the sunlight. Well, in this case, this is a different example, but in this example, what are the constants? Well, both groups are going to be given a substance so that we know it's not just the fact that they're getting a substance, it's that they're getting a different substance. So you always want to give both groups something, even if it's just a water pill or like a sugar pill, for example, or a water injection, so we can account for that placebo. But one of the things held constant here was that they were monitored for the same amount of time, and they should have been given the same number of doses and all of those things. So in this example, it says a scientist has 3 roses in separate but identical vases. Each rose receives the same amount of sunlight and water. So what are the constants? Sunlight, water, and then of course the vases. It says, but a teaspoon of pulverized aspirin is added to vase 1, a teaspoon of flour preservative is added to vase 2, and nothing is added to vase 3. Each rose was then observed, and the length of time that the rose stayed healthy with no sign of wilting or decay was recorded. So then you're trying to measure the effect that this pulverized aspirin in this case had on the vases, on the roses in the vases. So what are control variables? These are the variables that we don't want to change. We want them to be constant and the same across all groups. If you don't do that, then you can't really compare each group to one another. And not only do we have controlled variables where the water, the sunlight, the vase size, the soil quality, all that's the same, you also have one group that does not get the drug or get the fluoride, and we call that the control group. So the experimental group gets, the variable that we're trying to measure its effect, and the control group does not get that. That way we can compare that group to the control group. So it's used as a basis of comparison. A controlled variable is often used interchangeably with a constant, so we may use those words interchangeably, although in many cases they do represent different parts of the experiment. It just depends on how it's worded. You will be able to pick out what is what. A true control represents something located in the control group that is similar to the independent variable in the experimental group but has no effect. So the best example of a true control is a placebo. So for example, in that previous drug trial where one group got the new medication and the other group got the old medication. And it would probably be good to have a placebo group that takes the pill or takes the inhaler, but that inhaler doesn't have any chemicals in it. It's just a water vapor, or it's just a sugar pill. So that those people thinking they're taking the drug may cause them to have some positive results. Placebos usually have an effect. That's why they call it the placebo effect. It's just usually smaller than what an actual effective drug that's been designed for that to solve that problem will actually do. But sometimes they don't. Sometimes a placebo is just as good as the drug, in which case it shows that it's not actually the drug that is causing the effect. It's just that people think that taking this drug has helped them. So let's look at one last example. I believe this is the last example. So an experiment for a new foot fungus medication was set up. The medication was contained in a cream in which a new plant extract was added. There were 2 groups. Each group contained 25 individuals with foot fungus. Group 1 was given the cream, which contained the new plant extract. So this was a blue tube. While group 2 was given the same cream, but without the plant extract added. Each patient was also required to apply a specific amount of the cream, and each patient was asked to apply the cream once in the morning and once at night. So, what is the control group? Well, the experiment is for this new foot fungus medication. And the medication was contained in a cream in which a new plant extract was added. So what they're doing is they're seeing when they add this new plant extract, is this, helping this medication do better. So we have two groups. Group 1 is given the cream with the plant extract. Group 2 is given the cream without. So the control group is the group that's not given the plant extract. What are the constants? Well, what's held constant? It says that each patient was required to apply a specific amount. It should have been the same. Each patient is asked to apply it the same amount of times, once in the morning and once at night. So those are controlled variables, but the control group would be the group not given the plant extract. So in general, the larger number of individuals in the experiment, the more reliable the results. So the magic number from when I was doing some research and when I was leading my research teams in our science club was you really want a sample size of 30, but more is always better. If you had 1,000 people taking this medicine and 1,000 taking a placebo, that's going to give you better reliable results versus 20 people taking the medicine and 20 taking a placebo. Because then just random error could account for any differences, and you don't really know if the drug's actually helping. The number of individuals involved in the experiment is called the sample size. So you want to have large sample sizes because that can account for just like random errors and things that might cause one person to have crazy results. If you have 10 people, if you think about just an average height, and you're trying to figure out the average height of America, and you have 10 people, but the 10 people you select are college basketball players, and you base that to determine the average height of the average, American, you're going to think that they're way taller than they actually are because your sample size was so small and it was from a biased group of basketball players instead of just your average person. So you want to account for that. So at the end of these experiments, and some of the experiments that you will do, is you would write a conclusion. And conclusions are going to be based on, do you reject or accept, and usually you refer to the null hypothesis. So a rejected hypothesis is not necessarily a failure. It only means that there isn't a relationship between the independent and dependent variables, which is a good thing to figure out. Some of my students in the past, they're like, what do I do if my experiment doesn't show? One of the experiments we did was we put fluoride on seeds to see if it would affect their germination rate. And before we did it, we found out that it did. But before we did the experiment, they were like, what happens if, nothing happens? You write that in your paper. And that's good to know. You just learned something that this does not have an effect on plants. And that's good to know too. So it's just good to be honest about what you have and don't try to misrepresent it or create trends where there aren't trends. So the hypothesis that there is not a correlation or relationship between the independent and dependent variables, we call that a null hypothesis. So for example, with the fluoride on the plants example, a null hypothesis for that would be, if we add fluoride to plants, then it will not affect their plant growth. So the null hypothesis is always the prediction that there is not a relationship between these two things, between these two factors. So a theory would be a comprehensive unifying explanation about some aspect of the natural world. Theories come from following the scientific method over and over and over to reiterate some general, principle that we think might be true based on all the data that we've collected. So it's a comprehensive unifying explanation. So an example would be cell theory, right? All living things are made of cells. All cells come from pre-existing cells. The cell is the basic unit of life. So those three claims is what cell theory is. And we say it's a theory because it's a unifying explanation about the natural world. That all these living things, no matter what it is, if it's alive, it's made of cells. And our observations continually reaffirm that theory. Think about theory of evolution, just in terms of natural selection. Nature chooses who lives and who dies. Organisms with beneficial adaptations are more likely to survive and be selected by nature. And generally, that is true. So it's a theory that explains, animal behavior over a long period of time. Big Bang Theory, that the universe is expanding. Our observations of the universe show that it is expanding. If you backtrack that back, it would go into a single point, the center of the universe. So a lot of cosmological observations or astronomical observations support that theory. It's a unifying theory to explain the natural world. So theories are often modified with, I know one of the telescopes that came back with some new data made people start to question the Big Bang Theory, but then they realized that it actually did support it, but there's a lot of debate about some of those pictures. But the point is that these theories can be modified or changed. It's not something that's going to be true forever necessarily. It's just something that we think is correct. So it's what a scientist is most sure of, based on all the evidence and experimentation. So then what makes something alive? For something to be considered living, it has to have cellular organization. It has to be organized in cells, it has to be made of cells, with that organization of, if it's prokaryotes, it doesn't have organelles, but it has an organization that is cellular in its basic units. It has to have metabolism. It has to consume food. It has to eat and drink in some way to consume energy, to allow it to survive. Metabolism is just all those chemical reactions going on in the organism, or in that organism if it's one cell. Homeostasis, things that are living try to maintain some kind of stable internal environment. Living things have to reproduce. Otherwise, when they died off, they would just all be gone. So for something to be considered living, it has to reproduce. Heredity, it has to be able to pass on genetic information from one generation to the next, which ties into that reproduction. And then it has to be able to adapt to its environment. All living things adapt to their environment. So if something's alive, it exhibits these properties. So for cellular organization, specifically, every living thing is composed of cells. Now it might just be, if it's a bacteria, it's just a single cell, but it's still a cell. So if something is alive, it's going to be made of cells. Versus like a tree where billions, trillions of cells, but it's still made of cells. If something's made of only one cell, we say it is uni meaning one, cellular, one cell, unicellular. If something is made of more than one cell, we say it is multicellular. So for metabolism, that's the sum of all chemical reactions needed to sustain life. So in order to live, obviously you have to move, you have to break down food when you eat it, you have to be able to move oxygen to your brain. All of those things require energy, they require proteins. So we have to be able to make energy, and we have to be able to get that energy from what we eat. So all of that is metabolism, the sum of all of those things going on. Inputs and outputs is our metabolism. So all living things produce energy. All chemical reactions are a part of an organism's metabolism. And cellular respiration, when we get to that topic, that's going to be how we actually produce all of the ATP that we need to keep our bodies alive. And you'll notice that term ATP is going to come up a lot when we talk about energy. So certain organisms, we talk about homeostasis. So living things want to maintain homeostasis. Certain organisms can do that, and animals do that differently than others. One method of doing this would be to be able to consume lots of food and burn that food to produce heat to keep our bodies warm. We call these organisms endotherms. They produce a lot of heat because they eat a lot of food and they break down that food to produce heat. So endotherms, they maintain a nearly constant internal temperature, but they also have to eat a lot more food. So there's benefits and drawbacks to being an endotherm. They produce a lot of body heat, but they must eat a lot to generate that heat. And of course, they have adaptations that, as endotherms, allow them to help regulate their body temperature. So one of the things with humans, talk about mammals, birds, we sweat to help cool ourselves down. So we produce all this heat because we eat so much. But if we get too hot, then our bodies have this adaptation of sweat glands to help release a lot of that heat. So that would be endotherms. Eat a lot, produce a lot of heat. Ectotherms, these animals don't eat that much and they don't produce very much heat. So they have to find other ways to get their bodies warm. So you'll see them sunbathing. They'll go out in the sun. We think of turtles. We call these ectotherms because they have to get heat from outside of themselves. They have to go out and sunbathe or crawl into warm water, crawl into a cave somewhere to have a stable temperature. So they don't eat as much as endotherms. They have to get heat from their environment. In this case, we see this turtle basking. And they also use behaviors to cool themselves down. So you might see a turtle, burrow in the mud. So this would be like reptiles, amphibians, or fishes. So ectotherms have that lower metabolism, and they have to get that heat from outside of themselves. Endotherms have a higher metabolism because they generate their own heat from within themselves. So endo means within, thermic heat, inside heat. They're making their own heat. Ectotherms, outside heat. They have to go out and find the heat. Either way, both want to warm themselves up. So for endotherms, some examples will be birds and our mammals. So what happens when our bodies get out of homeostasis, when they get away from where our bodies want to be as far as temperature, pH, or whatever it is? So homeostasis is the ability of an organism to maintain a stable internal condition. So these mechanisms have to be automatic. We don't want to be thinking about having to regulate our body temperature. It would just be way too much information to process, and we wouldn't be able to consciously even do that. So these mechanisms are automatic. If we think about, when we get too hot, what do? Well, we sweat. We're out of homeostasis. We're too hot. We want to be in homeostasis. We want to be in that internal stable condition. But we get too hot, so our bodies sweat. That sweat produces liquid on the surface of our skin that allows the heat to escape into that liquid, and then it can evaporate. Our blood vessels might dilate. So that can bring blood closer to the surface. The blood has a lot of heat in it. So if the blood gets closer to the surface of our skin, then more heat will be released. If we get too cold, we shiver. And that shivering allows us to create movement, and it warms us up. And on the converse of when we're too hot, we have blood constriction. So this keeps blood vessel constriction. It keeps the blood away from the surface of our skin, more internal at our core, so that our core temperature can be maintained in homeostasis. That way, our major organs, our brain, our heart, and our lungs can be at the ideal temperature. Think about water balance. We want to maintain homeostasis with water and all of our electrolytes. So if you start to get dehydrated, your body responds by increasing thirst. That way you will go drink water to bring that water back into balance with your body. It might decrease saliva. That would make you thirstier and also conserve water. And it may concentrate your urine, which would also conserve water. So, you will need to memorize every one of these. Only joking. If you get all the way to anatomy 2, you actually will learn a lot of these, but it's just to show you that our bodies are constantly regulating our internal conditions, whether that's hormone levels, our oxygen in our blood, whatever it is, water, electrolytes, all of those things are being regulated to maintain balance or this ideal condition in our bodies. Why does our body do that? A huge reason for that depends on the example, but a big reason, if we think about temperature, is our proteins. Our proteins need to be at a certain temperature to function properly. If your proteins get too hot, they fall apart. And one of your most important proteins moves oxygen to your brain. So if your body gets too hot, that protein breaks down, and then you can't move oxygen to your brain, and then you're going to die. So all of these things actually keep you alive, and that's why it's so important that our bodies are able to do this. So reproduction is another quality of a living thing. So this would be the production of a new generation, producing offspring. So there's two types of reproduction. We have sexual and asexual. Sexual would mean you have two parents that when they have an offspring, sperm meets the egg, and you get a unique individual. So the offspring is a combination, and there's a genetic shuffling when the sperm and egg cells are made, which we'll get to. But the offspring are genetically unique. And that's a huge benefit to sexual reproduction, because having genetically unique offspring means that some offspring will have advantages over their competition. Some will have disadvantages, but some will have advantages, which will lead, if the environment's changing a lot, that will allow that species to survive as the environment changes. With asexual reproduction, we have one parent, and it just clones itself, just creates an exact copy. And this is what we see with bacteria. They don't find a mate. They just, double all their DNA and double their cytoplasm and their ribosomes, and then they just split in half. And then they have two of them. Now, the benefits to this is it's super fast, right? It's just making an exact copy. Bacteria are small. They don't have a lot of DNA. They can make an exact copy really quick. But the problem is, because the offspring look identical to the parents, it's hard for them to adapt to their environment. The environment changes, and all 10 billion bacteria have the exact same DNA. Well, if that environment's not favorable, then they're all going to die. But because they reproduce so fast, one of the ways that bacteria have overcome that disadvantage to all having the same DNA is that when your generation frequency is so fast, right, a new generation every 4 hours, well, in that case, mutations start to become so predominant, they're happening at such a high rate, that now mutations can lead to the adaptations that allow them to survive, rather than with sexual reproduction that have a longer generation time. In that case, it's more about the genetic shuffling that forms the sperm and egg cells than it is about mutation rates, although that still affects it. Heredity is something living things have in common, and that would be the transmission of traits from parent to offspring. So it's not just that they are reproducing, but it's that their traits are being passed down to their offspring. And this would occur through DNA. So your parents, the sperm met the egg cell, 23 chromosomes were transferred from your father, 23 chromosomes were transferred from your mother in her egg cell, and when those fuse together, you get back to 46, back to that original diploid number, and you are a unique combination of both your mother and your father. And it's not only that you got 23 chromosomes from your dad, it's that those 23 chromosomes themselves were mixed up and shuffled so that the chromosomes donated to you were unique. They weren't identical. It's not like half of your father's DNA. It's a shuffling of half of your father's DNA. And the same for the mother, so that combination becomes, you have the only DNA of its kind that will ever be, unless you have a twin, of course, an identical twin. But anyway, life evolves, so organisms adapt to their environment would be the next one there. And all organisms do this. And it can be a little confusing because you think that as an organism grows up, as you grow from an infant to an adult, that's not really evolution. When we think of evolution, we think of what happens to a population. Now that technically is change, but with evolution we're talking about changes in the genetics of the population is really how we measure it and the traits that those genes produce. So really that happens at the population level. Here they have an example of how organisms prehistoric to horses would have changed over time. So these organisms do change in their populations as offspring are produced. So you need multiple generations for evolution to really be measured. Now, what are some things that aren't living that might seem to have some of these qualities of living things? Well, viruses, at first they were debated whether or not they were a living thing, but the reason they are not considered to be alive is because they do not meet all of the qualities of living things. They are not organized by cells. They do not have a cellular organization, and they do not have metabolism. A virus, depending on which one it is, could be just left on the table for several days. It doesn't need any food. It could be preserved somewhere in a lab for forever because it doesn't have metabolism. And it's not cellular, so the same things that might degrade a natural cell tissue won't degrade a virus in the same way. So even though they may have some of the qualities of living things or seem to, they do not have all, so therefore they are not living. And then the last topic for this first set of notes, and those will be posted to Canvas, of course, is that life is highly organized in this hierarchy. So when you put together all of the things that a cell has, it has its nucleus, its mitochondria, its Golgi, and all of those cellular organelles, ribosomes, DNA, RNA, all those things. Each one of those things individually is not life, right? The mitochondria, the nucleus is not a living thing. But when you put the nucleus with the mitochondria and you have a cell membrane and you have all those other things, well then life emerges from it. So we say that life has emergent properties. When you put all these pieces together, you get life. The emergent property of the cellular level is life. So if we go below the cellular level, we would say that life does not exist. For example, the Golgi apparatus is not a living thing, but when combined with the other cellular components, you have life. All right, I think that's it. Here's just a little summary chart for you of the hierarchy of life. So it all starts at the atomic level. Atoms are made of these subatomic particles of protons, neutrons, electrons. And you could go, farther down than that to quarks or whatever. But basically you have the atoms. Atoms make up molecules. So I could take, an oxygen, put it with two hydrogens, and we get a water molecule. Then we could put some hydrogens and carbons together, and we get some lipids, which can make a cell membrane. And then we use that cell membrane, perhaps with some other cellular components, to make, an organelle. We get a mitochondria, which do have their own membranes. And then we take that mitochondria and add it to the Golgi and all the other cellular structures, and you get a cell. You put cells together, hundreds of muscle cells, thousands, and you get muscle tissues. You put muscle tissues together, and you get, organs. You get heart, which would be an organ. Then you put that heart with all the vascular tissue, and you get the circulatory system, which would be an organ system. You put the circulatory system with the nervous system and the muscular system, and you get the organism. So let's say we're talking about humans. Put many humans together, many organisms together, and you get a population of humans. Then you take the humans in wherever, Talladega, Raglan, and that population, all of them together become the species in the entire world. Then you could take all these different species together, how humans interact with birds and cows, and that becomes the community. Then when we take all of the different living species together and their natural environment, we get an ecosystem. And then we take that ecosystem, and these will also be on the notes as well that you can look at if the video hasn't helped you as much as you would like. But we take all the living things with their natural environment together, we get an ecosystem. Put all the different ecosystems or different ecosystems in an area together, you can get a biome, and then put a biome together, all the biomes on the planet, and you would get the biosphere, which would be the entire planet. Thank you for listening. That's the first notes for the introductory material. Hopefully I'll get a little bit faster at doing these as the year goes. You'll also have the notes on Canvas you can look at, and there'll be study guides, and there's quizzes. So what I would do is after you watch the videos, go ahead and take the quizzes so it's fresh on your mind.